Thank you, Andrew, and good morning, everybody. As you know, I am a pharmacologist, and I've worked for many years within the industry, and it's that experience in industry. I was 30 years at Glaxo, with 30 years of animal experimentation behind me. It's quite clear that it became increasingly clear to me, the problems that are involved in trying to extrapolate from any one species to any other species. And of course, what we're trying to do in the pharmaceutical industry is extrapolate to humans. And frankly, if you rely on animal data, you can get any results you want simply by choosing your species very carefully. Anyway, this morning, I had this... There's something I must do here to make this move. What don't I know? How about that? Yes. Right. The current approach that we have to identifying safe and effective new medicines is clearly failing. The industry is in great trouble. And coupled with this, of course, is an important need, or an urgent need for change. But to what? And how can we actually achieve it? And I would argue, I believe for a long time, that the only way forward is to focus our studies on human biology. We're hoping to treat humans, so why are we looking at dogs, rats, cats, mice, etc.? I truly believe this is the only logical way forward, and that we must increase our focus on the relevant species. But within this, of course, there's an important element or aspect of validation. And validation is something that really must occur before we can rely on any test. We must be certain that it's going to be of some value to us. And there are sort of two aspects to validation. One is the sort of technical side. How robust is it? How reproducible is it? How easy is it to transfer the technology from one centre to another? If you can't do that, then of course the fundamental properties of that new test are at question. But the other aspect, and that is something which really can be studied and is studied in official bodies, looking at these aspects to make sure that you do the same thing time and time again, you get the same results. But that's only one part of it, and the other part relates to the what I would term functional validation, which is predictive value, which is really ultimately the only thing that matters. Doesn't matter how robust it is, and it's got no predictive value, there's no point in doing it. So predictive value is very important. And predictive value can be broken down into its ability to identify potential toxicities, which we might regard as the sensitivity of the test, and its ability to identify lack of toxicity, which we regard as specificity of the test. There are many tests out there which those using them hold are very effective in identifying toxicities, because drugs, there seems to be reasonable concordance between toxicology in one species and another species, and where it has been identified also in humans. But it is only one aspect, and it could be that your test identifies, whatever it is, identifies every compound as toxic. So its sensitivity would be 100%. Brilliant test. Everything's toxic, don't test anything in man, we're safe. But that's not really much good for what we're actually trying to do, which is to identify safe, effective new medicines. So the specificity is an additional and equally important component. But how do you determine how good a test is? One of the problems we have is that if a drug proves toxic in a test species, the chances of it going into man are very limited. So what do we know about its toxicity in the relevant species? And the answer is very little. We are extrapolating, but who knows? It might be quite tolerable. And if you look at simple things like foods, there are very many foods that if they were subjected to the test methods that we currently use for drugs, would not pass. We would not be allowed to buy avocados and grapes and blue cheese and orange juice and coffee and Brussels sprouts. It goes on and on. Oh, and chocolate, of course. Chocolate, very nasty for dogs. So that sort of thing illustrates the uncertainty that we have in terms of the methods that we are currently applying. So what we really need to do is somehow find a way of validating our new approach. But this does present a significant problem, and the way that we at large tend to do this is to compare against an existing method. We say we've got to have an alternative approach here. Let's validate it against what we're currently using, which is not very good. We have to realise that few, if any, of the current animal-based methods that we're using has ever been formally validated. They're used. They've always been used. We rely on them. We have this fundamental belief that actually at the bottom of it all that they're pretty useful. They get it right quite a lot of the time. But they've never been validated. We don't know how good they are. We have no metrics at the moment that we can apply to existing methods to say it has this number or that number with this precision. Yet we are validating alternative methods against them. It really isn't very fair. An alternative method has very little chance of getting through if we are comparing something in a very rigorous fashion against something that's never ever been validated itself. So I would argue it doesn't really make a great deal of sense. I want to introduce the concept of likelihood ratios. Likelihood ratios were first introduced by Altman and Bland to apply to diagnostic tests. How valuable are diagnostic tests? How reliable are they? How useful are they? Well, they came up with this concept of likelihood ratios, which give you some metrics of predictive value. They take into account not only sort of ordinary sort of correspondence or concordance, but true positive data, false positive data, true negative data, and false negative data. And ultimately, these can be used to generate positive likelihood ratios and negative likelihood ratios, each of those providing a numerical score. And the positive likelihood ratio tells you the likelihood that a compound that's toxic in animals, for example, if applied to animal tests, is likely to be similarly toxic in humans. That's the sensitivity. And the negative likelihood ratio gives you the specificity in terms of its non-toxic profile. That's great. Now, here are some data that appeared in papers, a couple of papers by Jared Bailey and his colleagues, looking at the use of likelihood ratios in evaluating the use of dogs, rabbits, mice, and rats as species for identifying toxicity, potential human toxicity. They have sampled a wide range of compounds where data are available, and they are limited to those compounds where some data from animals and from humans on toxic profiles are present. Now that does limit the number of compounds, but there are very many compounds. Well, the numbers are actually included there. And what we have is the ratios that they have generated. Now, I can't tell you what these actual numbers mean, and in a way it doesn't really matter, except that the greater that number is than 1, the more confidence we have that it is indicating some, let's say, some relevance and some predictive value. And we see, for example, from the positive side, we have a value of 28 for the dog, looking at these various drugs. But the spread is huge. It goes from 4.7 up to over 500. The values generated for the other species are somewhat greater, with the rat giving us the biggest number of 253. But again, the range is absolutely huge. The confidence we have in those numbers is obviously limited, but nevertheless there is a number that has been generated. On the other hand, we look at the negative side, the information it gives us about non-toxicity, it is virtually absent. You cannot be certain that a drug that goes into an animal and comes out non-toxic is going to be non-toxic in humans. These values, they're all significantly greater than 1, but you can see that particularly for the dog and the rabbit, it's almost a technicality. There is almost no value generated by these species in telling you anything about non-toxicity. So, although I don't know exactly what those numbers mean, I can't explain exactly how good a value of 28 is or 256. The fact is, it is a number that has been generated by this process, and it gives us some level of quantification. Now, if we can then apply this process to these same drugs tested in using alternative approaches, human-based approaches, be they in vitro, in vivo, in silico, or a combination, then we would have numbers that we can compare with numbers. So we would have a clear idea whether we are actually generating any benefit by using this particular alternative approach. So direct, relevant, quantitative comparison. Then we're getting into science. We've got numbers, we've got mathematics. Now, I've just covered that there are ways of quantifying the value of these various tests, be they animal-based tests or be they human-based tests, and making some sort of comparison. And I do strongly believe that we should be applying such tests to evaluating alternatives. Because even if an alternative is not perfect, and no alternative test will be perfect, the question really is it better in scientific output than what we're currently using? Because ethically it's going to be. Ethically it's going to be. If it's at least as good in terms of its predictive value, then we should be using it on the basis that we should not be causing stress to animals unnecessarily. If there's a way of getting away from that, then it's the issue of don't do it. Use your alternative. Right. Now, I want to go on to an alternative approach, which we call pragmatic validation, which involves the use of existing data. And here, assessing the ability of human-based in vitro tests, and in silico, and in fact actually in vivo where possible, to detect toxicities missed by in vivo animal testing. We have data. There are many compounds that have been through the mill. They've been tested exhaustively in preclinical tests. They have proved themselves in those preclinical tests, be they the rodents and the non-rodents, to be effectively well tolerated. Therefore, they've been given a license to be marketed. They've gone onto the market, and then they have injured people. So here we have some good examples where there are drugs where the animals have not given us useful information. So we have gone out to identify a series of drugs where this is the case, clear examples where this is the case. For each of those compounds, we have then identified a partner compound. And that is a compound that is structurally and or functionally similar to the original compound, but doesn't exist the same toxicity. Or if it does, it's at a very much reduced level. So now we have a positive control and a negative control. Then, to submit these to a range of human-based technologies, again, be they in vitro, in silico, in vivo, whatever, submit them to those technologies and see whether any of those technologies, alone or in combination with others, could have identified the known toxicity, the toxicity that was actually experienced in the clinic. Because we have the data, we have the animal preclinical data, we have the clinical data. What we need to do now is to marry this up with further, as it were, preclinical data using human-based approaches to see whether they would have done better, and select those tests where the performance is increased. And this is just a schematic. So, marketed drugs that in preclinical terms were judged to be clean. Then, of course, we have the clinical outcome for those. Some of those will prove to be toxic, some non-toxic, or, in the case of non-toxic, less toxic. Submit those all to a range of tests, and then, in a blind fashion, analyse the data, see what comes out, and then compare that to what we know about the clinical profiles of those drugs. And some such studies have actually already been performed. And here is an example. This was something we published some years ago in my Pharmagene days. And this is looking at troglitazone, which is now very well accepted to be a rather toxic anti-diabetic drug, and rosiglitazone, which is not without problems itself, but in terms of the primary toxicity of troglitazone, which is liver toxicity, rosiglitazone is much less hepatotoxic than troglitazone, and it's whatever toxicity has lies elsewhere. And here we've looked at the effect on primary human hepatocytes. It's a very simple test, and we've compared the toxic outcome. The measure of toxicity is simply, in this particular case, ALT levels. So it's loss of ALT from these cells. It works with other markers, LDH, for example, get exactly the same results. And we have included in this the Cmax. Now, these are the maximum therapeutic blood levels expected or measured with therapeutic use of these compounds. And what we can see is that for troglitazone in the red, we get a remarkable increase in efflux of ALT at a level not very much greater than the Cmax, a very small therapeutic window of something like, according to that, something like threefold, not something that should be fast-tracked into the clinic. With rosiglitazone, it's quite different. Yes, it gives some indication of toxicity at a higher dose, but it is, in fact, in anti-diabetic activities, a bit more potent, and the therapeutic index is somewhere nearer to 100-fold. So here's a very simple test with two drugs for which we have clinical data, and this clearly at least accords with the data, the known clinical data that we have. So this very simple human hepatocyte test, had that been performed before troglitazone went on the market, it would have saved many lives. And here's another example, and again we're back with the glitazones, and here we've compared troglitazone and pioglitazone. These are results that I've been given by Ellen Berg at Discoverex using their biomap technology, and here this is a high content technology, and I don't know whether any of you are aware of this, but it's a very interesting technology where they look at a whole range of markers of stress and damage in response to applied drugs. And what you actually want to see in a clean drug is for the line to be absolutely straight, so nothing deviating from that line. Anything above is upregulated, anything below the line is downregulated. And what you can see here for pioglitazone is that going up to 100 micromolar, absolutely nothing changed. Whereas for troglitazone, our known toxic glitazone, you can see things are all over the place. And again, had that been performed before the drug went on the market, that would have been a really serious warning of the potential toxicity of this compound. So it's these sorts of tests that would be applied to the various compounds that I had previously identified. Now, we have a study underway. We initially, when we thought about this sort of approach, we identified 5 toxic, non-toxic pairs, so a total of 10 compounds, which had toxicities in a range of organ systems, and heart, liver, kidney, muscle. Interestingly, the drugs that we had identified were all present in ToxCast's phase 3 in vitro profiling platform. And ToxCast have agreed to provide us with the data. They have chosen the various assay systems, the tests that they're actually applying. We have no input to that, so we are completely at their mercy. They're going to test these compounds along with thousands of others, and for our compounds they'll give us prior look at the data, and we'll just see again whether our hypothesis holds up, that we can predict using the appropriate in vitro test systems, we can identify known toxicities. But whatever comes out of this, the outcome is to be published. Even if it doesn't support our position, the outcome has to be published, because negative data, of course, are just as valuable as positive. Having got that on the road, we then structured another study, which is on the same basis. But here, instead of just looking at a wide range of different toxicities, we decided to concentrate on liver and cardiovascular toxicities. A much wider range of compounds. In fact, as far as the liver is concerned, the FDA has a database of compounds actually mapping their or containing all their data on clinical toxicities and enabling us to do similarity searches. So it enables us to find compounds which are structurally similar and identify the toxicity associated with them, at least the hepatotoxicity. Again, then the question of performing that study and then analysing the data, and again, results be published and hopefully influencing in some way, if appropriate, regulatory guidance. That study was submitted for a recent funding call. Unfortunately, it wasn't successful, so we have a consortium of partners here who can undertake all the various parts of this particular study, awaiting a source of funding. So if anyone's got any spare millions, we'd be grateful to hear from you. And our immediate goals from all this are to obviously to analyse the data and to publish, whatever the outcome, and influence FDA and other regulatory guidance as appropriate. And to move on from there, if this is successful, to move on to look at other toxicities and perhaps to go into greater depth with the toxicities we've already studied. So that's that. I consider that this could be a very powerful way forward. Again, because we're looking at drugs where we know the clinical outcome, and one of the problems with evaluating existing methods is, by and large, we don't know the clinical outcome, except where animals told us, okay, this is safe to use. And we have, in some way, to move forward with evaluating, and if I can say validating, alternative methods, and validating them in isolation, and then comparing them back to unvalidated test methods is not a satisfactory way forward. But this is not the whole problem. There are further issues. Even if all this works out absolutely fine and we end up with great alternatives, and the regulatory authorities say, yep, these are great, and we're going to incorporate them in regulatory, insist on their incorporation in regulatory submissions, there are still problems to be overcome. Within the industry, there are such things as entrenched views and vested interest. I've always used this test. I'm pretty good at doing it. The last thing I want to do is to go and do something else. All my funding comes from my expertise in this test, so why would I then want to go and do something in which I've got no experience? Or complacency. It'll do. It picks up most toxicity. Yeah, don't worry about it. It'll all be okay. Or scepticism. I really don't trust these newfangled approaches. Yeah, it's human, but it's in vitro, it's in silico. How can that model what goes on in the whole integrated? No, I better keep using my rats and my dogs. Or lack of courage. There, it might cause delay in my regulatory submission. That could be extremely expensive, and that could count against me. Also, of course, if anything goes wrong with the drug that I've validated through this new approach, if anything goes wrong, could I be liable to litigation? Best stick with what we're already doing. So, those are problems. And here is a specific example, which I've been made aware of very recently, relating to the Drey's test, acute toxicity test for ocular and skin irritancy, developed in 70 years ago by John Drey's and his colleague Jacob Spines. Initially applied to cosmetics, but now applied to anything or since applied to obviously pharmaceuticals that were likely to come into contact with exterior surfaces, particularly the eye, the skin, and really a very unpleasant test, potentially extremely unpleasant, because it involves, certainly as far as the eye is concerned, administering the drug to the eye and then simply waiting to see what happens. And we all know how unpleasant something is when it's causing problems in your eye. Eye is extremely sensitive and extremely painful. And if you look at the results of some of these studies, and you can see a rabbit with a hugely inflamed eye, even an ulcerated eye, then it's not something that we should be doing. Fortunately, the Drey's test has been superseded. There are now a whole wide range of formally validated test methods for evaluating skin and eye irritation. The Drey's test is no longer required in the United States. In 2005, Abigail Jacobs of the FDA first publicly stated Drey's testing would no longer be required. And she's repeated this message subsequently. It is a consistent message. We do not do the Drey's test anymore. However, between the years of 2011 and 2014, of the 137 drugs approved in the US, 24% of them, essentially a quarter of them, were tested for skin and or eye irritancy. And despite the FDA's communication that Drey's is not required, 94% of all skin irritation and 60% of all eye irritation data included Drey's testing. Why? It wasn't necessary. Why did they do it? Worse than that, of these, 76% of the drugs were for systemic use. They weren't for topical use. What was the point of doing sensitivity testing on skin and eye for a drug that is going to be taken by mouth or intravenously? What is it about the Drey's testing that's so appealing that people just can't put it down? So, regulatory guidelines state that animal-based tests should not be used where non-animal alternatives exist. These are maybe not universal, but certainly in Europe and in the US, regulatory guidelines advise non-animal use where it is available against animal use. However, the researchers that I've just been talking about, and they come from all sorts of very well-known companies, well, not only ignored the guidelines requiring them not to use these animal-based methods, but even using them where they're not requested and not required. I think this is a very bizarre situation and something that really must be understood, addressed, and stopped. So, the message from my talk, I come to the end. We must continue to refine and develop human-based tests. We must explore and develop improved ways of validating those tests. We must lower the barrier to alternatives. It's too high at the moment, far too high. We must educate researchers in the value of these, on the existence and the value of these tests. We must enforce official guidelines. If the regulators say don't use a test, and you do use it, you should be taken to one side and dealt with. And we should set timelines. There's nothing like having a deadline to concentrate the mind. So, as with the REACH initiative, banning animal use in testing chemicals, that sort of approach should be taken for pharmaceuticals, where we say, right, you've got a finite amount of time in which to stop using these tests. It may be all tests, it may be some tests, but finite deadlines are key. There's nothing like them for focusing the mind and allowing ingenuity to be exposed and to be exploited. And I'd just like to thank various collaborators, Kathy Archibald and Katia Sayun, colleagues at Safer Medicines, Tammy Drake, colleague at Centre for Responsible Science in the US, Ellen Berg of DiscoverX, and Amanda Woodruff at Astram Bioscience and was Pharmagene And with that, I will say thank you very much for listening.